Okay, good morning. This is day 5. So as of now, you've taken 5 practice exams. I know on the screen it says day 7. Ignore that. I forgot to change it. But it is, in fact, day 5. All right, let's get going. Last one of session 1. Rank the following biological molecules in order of how readily they diffuse across the plasma membrane from the most diffusible to the least diffusible. So here, remember that the rate of diffusion highly depends on the hydrophobicity, charge, and size of a molecule. Ions in big polar molecules do not diffuse through the membrane, but instead are transported through channels or carrier proteins. Of all the molecules, CO2 is going to be the smallest, and therefore it'll diffuse the most rapidly. Glycerol has only 3 carbon atoms and is therefore the second smallest molecule. Note that the chlorine is small, but it's charged, and thus it'll need a channel to get through, right? An ion-gated channel often. So sucrose then, right, is actually a pretty big molecule. But it's not charged, so it'll be transported at the same rate than something like a chloride ion. So then, right, with that in mind, since we're ranking it from how readily, from most diffusible to least, so CO2 will be the most, right? And then it'll be glycerol, because that's a hydrocarbon, basically. And then it'll be sucrose, and then it'll be ions. And if you need to review this, it's chapter 7 in Campbell. Okay, you perform a Western blot on 2 proteins of similar molecular weights, about 50 kilodaltons, and you find only one band developed on your SDS page gel. How would you modify your assay to distinguish the 2 proteins? So in this case, right, statement C is correct because we have 2 proteins with similar size, and we need to examine other properties of the proteins in order to distinguish them. Proteins of the same size can have a different charge, and thus separating them isoelectrically may allow us to characterize them better. And if you need some more information on this, you can see the notes on this slide. And then, always email with questions if you still have them. And this is just a diagram of the Western blot. So if you needed some more information or to refresh your memory, you can get some more info here at this website. 3, the tiny hairs on a gecko's toes enable it to climb walls. The hairs are made of hydrophobic keratin and adhere to surfaces via van der Waals interactions. At the tiny interface where the gecko's toe hairs touch the surface it climbs, which amino acid are you least likely to find? So since we're told that the gecko's toe hairs are made of hydrophobic keratin, that means that there are plenty of hydrophobic amino acids. If you look at A, B, C, and D, you can see that all of them have hydrophobic side chains. In contrast, E, serine, its side chain is polar due to that OH group that we see. And so here, serine is the least likely amino acid to be observed in the toe hairs. And here's where you can get some more information if you need it. When found on the extracellular side of the cell, which of the following classes of membrane molecules is a marker for the phagocytosis of apoptotic cells? If you remember that phosphatidylserine, so I don't butcher it, I'll say it slow, is a common phospholipid found in the bilayer. And so it's usually held facing the cytosolic side of the plasma membrane by an enzyme called flippase. But this is super different from a typical phospholipid, which can freely flip between the two faces of the membrane. And so when a cell undergoes apoptosis, the phosphatidylserine is no longer restricted to the cytosolic domain, but instead flips to the outside of the plasma membrane, the extracellular side. So there they serve as a signal for macrophages to engulf cells undergoing apoptosis. And so if you go down the options, right, glycolipids are lipids with attached carbohydrate chains, and they provide energy to the cells and also serve as markers for cell-to-cell recognition. Think of like your blood types and things of that nature. They're important in those. And then sphingomyelin, or SM for short, is found in plasma membranes, but it's abundant in the myelin sheath that surrounds axons of nerve cells. And then PC at the end, so phosphatidylcholine, is a type of phospholipid found in membranes. It can be found in the inner leaflet of the lipid bilayer, similar to PS or phosphatidylserine. But the phosphatidylcholine is actually more abundant in nerve tissue in the white matter of the brain. So for all of these reasons, D is our correct answer. And again, all of this, basically everything I'm saying is in the notes. So if you need to go back to it, or these extra resources that are built in. 5, the presence of which of the following amino acids in the channel region of aquaporins contributes to the electrostatic selectivity to allow water, but not other molecules to pass through. So again, here, right, you've got to remember your chemical properties of your amino acids. So in this case, we have 2 asparaginines. I always say asparaginine. Asparagines they're involved in moving water through. Asparagine is the only polar amino acid that's shown here, or that you have the option of selecting. And because, water with our cohesive and adhesive properties, right, they move best and interact best with other polar molecules. And so again, this one, if you know that A, B, D, and E are not polar, then that'll help you find the answer to this one real quick, which is C. While preparing to teach an organic chemistry class, Professor Dog has managed to mix up several beakers. He has taken samples from each beaker and enlisted your help in identifying them. Below are the results of a series of reagent tests. This is something where you just have to know what each of these, the Biuret tests, right, that actually tests for peptides, Benedict's test for sugars, simple carbohydrates, Sudan tests for lipids, and the iodine tests for complex carbohydrates. Ninhydrin tests for amino acids. And so if you look, right, one thing that we've got going for us in this situation anyway, is that we have the Biuret test is different for one compared to the other 4. And so because of the fact that our Biuret solution is purple in number 3, we know we're looking for a protein. And that tells us, right, that we're dealing with an amino acid right there for 3 first. So I'm going to go back. So a solution of asparagine, right, that would turn purple because there are going to be peptide bonds formed there. And you would also see then pale yellow for the ninhydrin, right, if we're talking about this. And then 2 triglycerides. So basically, right, in 1, oh, sorry, I totally screwed this up. I'm gonna reset, but not really. Okay, kind of, and also not kind of at the same time. So each beaker, right, we've got to think of which each one tests for. And so if you go through, right, and just remember what color you would see for each given what, that will help you get to the right answer. I'm not going to butcher this anymore. I'm just going to move on. But if you have questions or need clarification on it, definitely ask me at 1 o'clock. 7, there are some rare organisms that do not have lipid bilayers, but instead have lipid monolayers. Which of these is a plausible structure for the lipids they use to make up their lipid monolayers? So in some archaea, the lipid bilayer is replaced by a lipid monolayer, in which tails of phospholipid molecules are connected into a single molecule with 2 polar heads, like this one. This structure makes their membranes more rigid compared to fluid bilayers, meaning that they're better able to resist harsh environments, like high temperature, and those types of things. Additionally, in A, you can also see the ether linkages, which is another characteristic of things like archaea. Number 8, one of your classmates has symptoms of protein deficiency. You suspect that she might have a mineral deficiency due to her diet of nothing but canned spaghetti. Which of the following minerals should you test for first? So proteins are really the only ones that contain, out of the only macromolecules, right? If you think of your carbohydrates, proteins, lipids, and nucleic acids, right? Proteins are the only ones that actually contain sulfur, and specifically in these 2 amino acids, methionine and cysteine. And so if you test for the presence of sulfur, it would help distinguish proteins. But you know, iron and copper and manganese and molybdenum, those are cofactors or coenzymes for other things. 9, a new therapeutic drug can treat the symptoms of the demyelinating disease, multiple sclerosis, by blockage of voltage-gated potassium channels. Neuron action potential conduction improves because right? If you remember, the voltage-gated potassium channel enables the efflux of potassium out of the cells. And so since a positive charge is leaving the cell, the cytoplasm becomes more negative, which means the cell is repolarizing. So because of that, an action potential subsides and the cell reestablishes a resting membrane potential. When the voltage-gated potassium channels are blocked, less potassium is transported out of the cell and more potassium stays inside the cells. This makes sorry, but anyway, and stays inside the cell. So this makes the cytoplasm more positive, and depolarization prolongs the action potential. So if we go through the other options, right, A is incorrect because the voltage-gated potassium channels increase membrane permeability to potassium and lead to potassium efflux, not influx. B is incorrect because the resting leak of potassium ions is regulated by non-gated potassium channels and not by voltage-gated potassium channels. Statement D is incorrect because an amplitude of an action potential is constant. And so it does not change because the action potentials are all or none events. And then E is incorrect because action potentials do not vary in amplitude. Okay. And then if you need more of a review, check the notes of this slide, as well as going back to chapter 48 in Campbell. Number 10, which component of Fick's law of diffusion is most optimized in the ventilation of fish gills compared to mammalian lungs? So the correct answer is B, the gradient for diffusion, because the fish create this high concentration gradient via the mechanism called countercurrent exchange, which maintains constant and high gradient for diffusion across the entire surface of the gills. So the gills of the fish are compromised of the lamella, which are very well vascularized, so they're comprised of the lamella. I don't know if I said compromised, that's what I felt like I said. Anyway, comprised of lamella, right? So, and that's what we see right here. And this is figure 42, so chapter 42 in Campbell is where you can brush up on this material. So this is one where the most optimized, right, is important. A isn't super relevant here, neither is D or E. You might get thrown by the C option, surface area, but remember, if we want to talk about surface area, it's got to be surface area to volume ratio. And so definitely the most optimized would be this gradient for diffusion. Pollen grains were treated with colchicine in culture. The resulting plants were, okay, so in chapter 15 of Campbell, it talks about this. So pollen grains are haploid, right? Because they're a product of meiosis. Colchicine is actually used to produce polyploid plants. Oftentimes, this is for basically taking something that's infertile and making it fertile again. So taking a triploid plant and making it hexaploid, for example. So this means that so if colchicine is in the mix, then that inhibits the microtubule formation during meiosis. And so what that means is that the chromosomes are not pulled apart. And so then you have cells that are now double the number of chromosomes that it would have normally had. So for example, right, seedless watermelons or bananas, both of these things, they are normally, if you get a wild one, they will produce seeds because they actually have an even number of chromosomes. But in order to make them seedless for our convenience, they make them triploid and then they are infertile. And colchicine can actually reverse that because it's like, well, if you make a lineage of infertile plants, how do we get more bananas and more seedless watermelons, right? Well, if you make them all infertile, you can't. And so you can reverse this infertility by applying colchicine to double their number of chromosomes again. And so because of that, B is the correct answer. So they're dihaploid. So now they've got two copies of their haploid or haploid genome, and now they're fertile. C is the kind of the opposite. Dihaploid is still the same, obviously, but sterile. And haploid and sterile is what colchicine will undo, right? And then haploid and fertile, you're not going to be haploid and fertile. So yeah, so basically C and E are kind of backwards of what would actually happen. D is actually the effect that we're undoing with colchicine, and B is the result of what happens to a haploid sterile plant in the presence of colchicine. So if you want to review this, like I said, chapter 15 in Campbell or chapter 11 in Raven also talks about polyploids. Okay, which of the following statements about seeds is false? Coleorhizae are found in monocotyledonous plants. So that's true. And here's figure, from chapter 38 in Campbell, figure 38.8. B, both monocots and dicots have radicals. Yes, that's true. They're very similar, right? So here's your radical in this one. Here it is in this one. C, monocots and dicots have a pericarp. It's true. So they do have the pericarp fused with a seed coat. And it's part of the fruit that develops from the ovary wall of the flower. So both will have that. D is correct because the coleoptile is the pointed protective sheath covering the emerging shoot only in monocots. And so E, the alurone layer, is found in both monocot and dicot plants. So it's not only found in dicotyledonous. So E is actually false. So if you remember, right, we had another question previously. Maybe it was from yesterday's exam or it was earlier in this one. They're blurring together. I can't remember. But you had a question about the importance of the alurone layer, right? And that's the layer that actually gets broken down to feed the embryo. And so it would make sense that both monocots and dicots had that, right? And not just dicots. All right, and again, this is the same figure that we had for the other one from chapter 39, that other question. Oh, I'm going backwards somehow. Which of the following terms best describes the closing of leaves or flowers at night? This is something that, you kind of just have to use your words here, right? So nasty, the ending, right? Nasty means movement, okay? So what do they move in response to? E is touch, thigmo is touch. You know, these mimosa plants actually shown here are sensitive to touch. You can actually see these, they grow on the sides of sidewalks here in Austin a lot. So I urge you, if you see something that looks like this, pet its leaves and they'll all close up. That's thigmonasty. Thermonasty would be temperature, right? Movement in response to temperature. Seismo, think seismograph, right? For earthquakes. Um, so that would be movement in response to like vibrations, right? B, nyctinasty is actually, I always think nicked like nighttime, right? So this is actually the correct answer. And then epinasty is actually more of a response that's induced by ethylene. And it really happens when only the upper part of the plant grows. And it often happens, so epi means above or on top, right? So epinasty is when just the top of the plant grows. And it tends to happen in response to flooding. Okay, so B is the correct answer here. And if you need to go back to it, right, chapter 28 in Raven is where you can find this information. And I know it's in Campbell too, but right off the top of my head, I don't remember exactly where it is. But here's the table from Raven. Okay, 14. Which of the following statements about carnivorous plants is false? All carnivorous plants are angiosperms. All carnivorous plants are evolved for nutrient poor environments. Okay, these sound right. C, all carnivorous plants are limited to small arthropods in the case of bladderwort, zooplankton, or in the case of bladderwort, zooplankton is prey. No, some of the, well, let's read all of these and let's see. So D, all carnivorous plants have mechanisms to attract and capture prey. Yes. E, all carnivorous plants derive energy from trapping and consuming arthropods and or zooplankton. So that's true, right? So actually the one that is incorrect here is C, and that's incorrect because they also capture other small animals. So there are some that, you know, eat things like protozoa, right, which are much smaller than arthropods. They can eat arthropods, but I've also heard of them ingesting things like mice. So, you know, they're not limited to just small arthropods or zooplankton. I think it's much broader than that. And even the bladderwort is not limited to just zooplankton, so they can eat tadpoles and little worms and all kinds of stuff. So C is our incorrect one. And the rest of this, right, A is something that you just kind of have to be familiar with. But the rest of this is really about how they have evolved, right, for this lifestyle. Whereas C is a little bit too, I'm not sure if specific is the right word, but it's really the only one that isn't talking about the evolutionary or adaptations, right, for this lifestyle. And so it kind of sticks out like a sore thumb. 15, roots absorb cations from the soil by a process called, so in this case, you know, soils are comprised of a bunch of different things, right? And all of those parts in soils tend to have a net negative charge. And so that's what we're seeing here. This is from Campbell chapter 37. And so because of that negative charge, they attract and retain positively charged particles. And at the same time, they repel other negatively charged particles. So you can see here the positive charges interacting with the negative charges. And so that means that the anions, right, are much more available to plants than the cations. And so mineral cations become available to the plant by cation exchange, which is a process where cations are displaced by other cations, such as like protons, right, from the soil particles and enter the soil solution. And so that's what this figure is actually showing you, is just the mechanism of cation exchange, which is our correct answer here. Which of the following statements about plant stomata is true? Here you've also actually seen a very similar question to this before, where the correct answer was similar. So I feel like we've gone over this. These question options are slightly different, but E is not, and I remember talking about this one, right? CO2 is not the only signal, right, that contributes to stomatal opening. But we've talked about D, and we even use the same figure. So I'm not going to go into length or go at length for this one. But in Campbell, you can check chapter 36 and also check the notes of this slide. 17, which of the following features of flowers is not meant to facilitate its pollination by animals? And so which of the following is not meant to facilitate? So sepals, right, are normally, let's see, so the correct answer, right, the presence of leaf-like sepals. You know, sepals are normally green, which means they're pretty boring considering all the other characteristics that flowers have, right? And if we're thinking about pollination, we're thinking about attraction, right? We want to attract things. So all these other options, A, the nectar at the base, right? That gives like a reward, right, to anything that comes and pollinates that flower. C, brightly colored and conspicuous petals. We've learned like that is an adaptation to attract pollinators. D, a strong odor of rotting meat, like right here. You know, some things like certain beetles and flies, they love that smell. So yeah, that fits as an adaptation. E, patterns only visible under UV, also a relevant choice here. So really the sepals are the only like kind of boring part of the plant. You know, they're just green hanging out underneath the petals. And so they're not meant to facilitate pollination. All right, the food source of Rhizopus, it's like bread mold, right? So if your bread on your counter gets moldy, that's what it is. The food source of Rhizopus has been depleted. Which of these is most likely to happen? This is familiar because we had another question. I can't remember what test it was on this week, but it was the one about the phytoplankton that switched to sexual reproduction from asexual reproduction, right? When the nutrient conditions were low in the water. I think in the water. Oh, excuse me. So here it's the same thing. You got to think about how can they recombine to get more genetic diversity if a food source is being depleted. And so the way that they do this, right, is in this case, you get mating types that fuse. Now, in fungi, right, they don't have female male, they have plus minus mating types instead. So that's why I'm saying mating types. And so, but when they fuse, that's called a zygosporangia or a zygosporangium. And so that will actually contain several haploid nuclei, which will later fuse, creating a diploid nuclei with some genetic diversity to it, right? So A is the correct answer here. B is incorrect because asexual reproduction occurs when conditions are favorable, right? We've talked about that before. C is incorrect because conidia are produced during asexual reproduction in Ascomycetes. Mycetes. D is incorrect because rhizopus is commonly found on the surface of bread. It's not a mycorrhizal fungus. E is incorrect because only fungi which have flagella are chytrids. And chytrids actually are the simplest fungi, but there's a group of them that infects amphibians and not your counterbred. All right, and if you need a refresher on the life cycle of the fungus, then chapter 31 in Campbell is your best bet. Stressful stimuli cause a number of physiological reactions. Which of the following is not a correctly matched statement about stress-related hormones? Okay, epinephrine stimulate glucose production from glycogen. That's correct because, right, in fight or flight, you need glucose, and glycogen is your storage molecule. And so you need to break off, these glucose molecules in order to, power fight or flight, right? B is correct because mineral corticoids increase blood pressure and volume. And so typically, so that's correct. Statement C is correct because glucocorticoids, like cortisol, reduce immune system activity and suppress production of white blood cells. D is correct because norepinephrine, similar to A, and the effects of adrenaline act in fight or flight, which increases your breathing rate. And so that leaves us with E. So the adrenocorticotropic hormone is a hormone produced in the pituitary gland, and it's produced in response to chronic stress. And its principal function is actually to trigger the release of corticosteroids from the adrenal cortex. And so that comes directly from presynaptic neurons, and it's not under hormonal activation. Again, check the notes section of this and chapter 45 in Campbell if you need to review. Which of the following hormones is not secreted by the anterior pituitary gland? So in this case, right, it's a matter of just understanding what's produced where. C is our correct answer here. And here we can see from chapter 45 in Campbell, what types of things are produced or synthesized in the hypothalamus. Sorry, I keep yawning. Okay, 21. A patient presents the following symptoms: diarrhea, dermatitis, and dementia, as well as necklace lesions on the lower neck, hyperpigmentation, thickening of the skin, inflammation of the mouth and tongue, digestive disturbances, amnesia, and delirium. Which of the following is deficient in this person's system? So in this case, right, this sounds like pellagra, which is a B3 deficiency disease, and it causes dementia, diarrhea, and all these things. So other vitamin deficiencies, if you're deficient in vitamin D, for example, you'll have rickets. If you have a vitamin C deficiency, you'll have scurvy. If you have a vitamin B1 deficiency, you'll have beriberi. And then you also, for vitamin E, right, for vitamin E deficiency, you would have some other symptoms. And to review this, right, you can go to this chart from chapter 41 in Campbell. So here, right, for vitamin E, since I just like left that sentence hanging, you'd have nervous system degeneration, vitamin K, right, defective blood clotting, so on and so forth. So this is a matter of just remembering, right, which things cause which symptoms, or which deficiencies of which things cause which symptoms. 22, which of the following membranes is or are responsible for respiration in developing birds and reptiles? So here we go with our amniotic egg. If you think about, you basically just have to remember the parts and what they do. So here, excuse me, the amnion, right, protects the embryo in a fluid-filled cavity that cushions against mechanical shock. The allantois is the disposable sac for metabolic wastes. The chorion is the membrane of the allantois that exchanges gas, right? So both of these are going to be your correct one, and those are B and C. So E is your correct answer here. What is the primary source of food for developing zebrafish larvae up to 5 days post-fertilization? So here, that's going to be the yolk sac, right? I mean, larval food, fish food flakes, if they're in an egg, right, they're not going to be able to get that. C, no food required. Of course, they require food. Even dormant plants, right, require food. That's why we have the alurone layer for them, which is similar to the yolk here. And then D, paramecium is a protist, which has nothing, well, that would be similar to having larval fish food flakes, right? But they're in an egg, so they don't have access to B or D. And they definitely require nutrients. And so, because they're still developing. So A, yolk sac is the correct answer. Let me see if okay. Okay, the virus, I'm not gonna try to butcher that right there. Oh, I just made this up. The virus, I just made this up, causes the following symptoms: overly fragile bones, fatigue, bone pain, an abnormal EKG with short QT interval and a long T wave, kidney stones, constipation, and vomiting. You theorize that the virus is infecting endocrine cells and causing the overproduction of a certain hormone. Which hormone should you test for first? So figure 4518, so this is chapter 45 in Campbell. This will remind you, right? So of the given options, only the parathyroid hormone has a direct effect on bones, and that's why it's our correct answer. But if you think about these other options, right, prolactin is produced in the anterior pituitary gland and it triggers the production of milk. Triiodothyronine is produced in the thyroid gland and it increases the metabolic rate and body temperature. D, melatonin is secreted from the pineal gland and plays an important role in circadian rhythms. And oxytocin is produced in the posterior pituitary gland and it triggers the release of milk. Um, so, for these reasons, B is the correct answer. And again, if you need to review, it's here. And again, it's in the notes of this slide as well, and on the slide also. 25, the flippers of dolphins are exposed to cold water away from the main mass of the body and employ countercurrent heat exchange. Which of the following statements are false? We've actually seen a question like this, but it was about penguins, if you remember, and we used the ducks as the example. And so here we can go one by one. So statement A, each artery in the flipper is surrounded by several veins, allowing efficient heat exchange. And that's correct because the arteries, which bring warm blood, are usually surrounded by veins, which are colder. Therefore, some heat from the blood traveling through the arteries is transferred to the venous blood rather than the environment. That's countercurrent heat exchange. B, as long as there is a difference in temperatures, heat transfer occurs from the warmer vessel to the cooler one, right? And so B is correct because, right, the second law of thermodynamics states that heat flows naturally from an object with higher temperature to an object with colder temperature. You could think about this, right, in terms of diffusion almost, right, from high concentration of energy, heat, to a low concentration of energy, less heat. C, the coolest blood is found at the tip of the dolphin flipper. That's true because the tip of the dolphin flipper is the most distant part of the body. Just like if you're somebody that's got cold toes and cold fingers a lot, like me, right? We know that's true. Dolphins are the only mammals known to employ countercurrent heat exchange. Well, we know that's actually false since we just talked about how penguins also do that, right? Ducks also do that, and whales, right? And other aquatic birds that I probably can't think of right now. So this is another one where we see that only word, right? And that's our red flag. So there are very few things that are only, right, in biology, or always, or, those types of things, or never. Okay, so E, once the arterial blood has been cooled past a certain point, it will no longer transfer heat to the veins. And so E is correct because when the temperature reaches equilibrium, right, there's no gradient anymore. So D is our best answer here, our false answer. And again, here's the same figure that you've seen before from chapter 40 in Campbell. 26, the diagram above depicts developing human sperm cells. We could probably knock that off because of the fact that you would see flagella, right, on a human sperm cell image. B, human blood smear. Well, let's see. If it was a human blood smear, there would be, anyway, no nuclei in there. Because if you remember, mammals don't have nuclei in their blood cells. C, human lung section. You would think that they wouldn't be so separate, right? You'd have a lot more of a tissue layer look as opposed to individual cells. See, frog eggs are actually pretty ginormous. And so here we're actually looking at a frog blood smear. Frog eggs you can see without a microscope, right? So you knock that one out. 27, which statement best explains why the ascending limb of the loop of Henle is thicker than the descending limb? So in this one, you have to remember that the ascending loop of Henle contains the triple symporter, which is the most active pump in the kidney nephron. So ions are charged particles, and energy in the form of ATP is needed to carry them across the membrane. ATP is synthesized in the mitochondria. The more mitochondria a cell has, the higher the metabolic rate of the cells, which means there's enough supply of energy for the constant transport of ions. However, in the descending loop of Henle is involved with water reabsorption. So since water is a small molecule that can freely diffuse across the membrane, then no energy is required for transport, even if they're going through, if water molecules are going through aquaporins, right? It's still just passive transport, not active. And so statement A is incorrect because aquaporins are found in the descending loop, and the ascending loop does not reabsorb water. Statement B is incorrect because both loops of Henle receive the same blood supply from the vasa recta. Statement D is incorrect because unexplained proliferation of cells may indicate a pathological process. And statement E is incorrect because the structure of the loop of Henle is not a vestige. It's an example of an evolutionary process that results in higher survival rates. So a vestigial feature would be like, right, your wisdom teeth or your appendix or something that doesn't really have a role anymore. It did at one point, but you know, way back in evolutionary time, but not anymore. So that's why C is a correct answer. And if you need to review this, oops, so sorry, you can go to chapter 44 in the Campbell book. 28, a child has overdosed on acetaminophen, causing acute liver failure. He displays symptoms of severe jaundice and swelling of the abdomen. Which of the following processes is not directly impacted? So erythropoietin synthesis takes place in the kidneys and not in the liver. And so its target organ is the bone marrow, where it actually induces the production of red blood cells. So erythropoietin is secreted when tissue oxygen concentration drops and there's a reduced capacity of oxygen delivery to cells. Therapeutically, it's used to correct anemia. Bile synthesis occurs in the liver, not in the gallbladder. So this process will be affected. The bilirubin glucuronidation, sorry, I haven't seen that word in a really long time. But anyway, those actually, so that adds the glucuronic acid to bilirubin, and that takes place in the liver. Production of serum albumin also occurs in the liver because it's the major organ which synthesizes almost all plasma proteins. And then glycogenesis occurs in the liver too. That's where we store our glycogen. And so if you need to check this out, right, chapter 42 in Campbell is where you can go for this. And D is our correct answer here because this will not be directly impacted by liver failure, right? Because it occurs in the kidneys. Fixation is a lab procedure used to prevent biological tissues from decay. In instances where perfect morphology of the whole animal is desired, cardiac fixation is the preferred method. Where should the researcher inject the fixative to ensure the best results? And so the left ventricle opens into the aorta, which directs all the blood to the systemic circulation to the entire body, right? Which is what goes to the entire body. So if the drug is injected into the left ventricle, it should reach every single body tissue. A and C are incorrect because the fixative will reach only the lungs and then will come back to the heart because the right side of the heart directs blood to the pulmonary circulation. B is incorrect because only vessels in the abdominal cavity will stay along with parts of the inferior vena cava and the right atrium. And statement D isn't quite right because the stain will be transported to the left ventricle and from the ventricle to the systemic circulation. And again, right, this is Campbell chapter 42 if you need to review this material. 30. What is the primary mechanism by which ADH exerts its effects on the kidney? So ADH is synthesized in the hypothalamus and stored to the posterior pituitary. It's also a peptide hormone that's produced in the supraoptic and paraventricular nuclei. And it's got two major types of vasopressin receptors, V1 and V2. V1 is found on blood vessels where it triggers vasoconstriction, and V2 is found in the basolateral membrane of the collecting tubules of the kidney. And so the action at the V2 receptor initiates a signaling cascade which leads to the activation of adenyl cyclase, which produces cyclic AMP. This causes aquaporin containing vesicles in the cytoplasm to move to and fuse with the apical membrane of the collecting tubule. And then, as a result, the membrane becomes permeable to water. Then water can move from the kidney into the nephron, and this conserves water. And so basically, ADH regulates water permeability, right? And so statement A, so that's where we get D, right? Statement A is incorrect because podocytes, along with the glomerular endothelium and the basal membrane from the filtration barrier, are not directly involved in the regulation of water reabsorption. So they really just act as a sieve. Statement B is incorrect because only vasoconstrictors can decrease the flow through the afferent atrial. Statement C is incorrect because elevated osmolarity in the medulla is regulated by reabsorption of salts from the ascending loop of Henle, which lacks ADH receptors. Statement E is incorrect because ADH targets water channels and not ion channels. And if you need some more info, you can go to this website from Colorado State that has a good, some good information there. Question 31. In the picture below, taken from the work of N. Tinbergen, a cardinal feeds minnows, which rose to the surface looking for food. The bird fed the fish for several weeks, probably because its nest had been destroyed. The cardinal's behavior is best understood as C, a fixed action pattern. What probably happened here is that having lost its offspring, the bird inadequately responded to a dominant parental instinct, which basically is a sight, it sees a small mouth open, eliciting, a stereotypic behavior, which is a fixed action pattern. Now, these are genetically determined. So for a fixed action pattern to be manifested, there must be a sign stimulus that triggers such a stereotypic behavior. In this case, it was probably the fish opening its mouth. Habituation is actually a form of learning, which an organism decreases or ceases to respond to a stimulus after some time if the stimulus is repeatedly presented, right? So you kind of get burned out, right, on the stimulus, for lack of a better word. Imprinting is a combination of innate and acquired behaviors that are usually released in response to a learned stimulus. So most imprinting promotes survival of newborn animals and shapes their future breeding activities. So these imprinting typically takes place during a certain sensitive period of postnatal life. So like in ducks and geese, right? They've got a 24 to 48 hour period after hatching when the mother following response is learned. When you see all these little ducklings following the mother duck, they learn that in the first 24 to 48 hours. And if you miss that period, right? Or that's also why you've heard probably of people having ducks follow them, right? It could be that the mother wasn't around in that 24 to 48 hour period, and but this person was there, right? And so the duck imprints on the person and follows them around like it would a mama duck. D, associative learning is actually the process by which an association between a stimuli or a behavior and a stimulus is learned. So that's things like Pavlov's dog, right, with conditional classical conditioning or operant conditioning. And then, right, operant conditioning is when a particular behavior is reinforced or punished, which results in an altered probability that the behavior will happen again. So the FAP, or the fixed action pattern, is really our best response here. And if you need to review this, right, you can go back, and we've talked about a similar one, right? We saw the sticklebacks in another question earlier this week. You can go to chapter 51 in the Campbell textbook. 32, which of the following is an example of habituation, right? So we just went through all of these examples. So if you remember, I just had said habituation is a form of learning in which an organism stops responding to a particular stimulus after repeated presentation. So let's look, see which option is best. So statement A is incorrect because it's an example of social learning, right? Many animals are capable of learning by observing other animals, and they frequently acquire foraging information and skills, learn to identify predators, learn calls, learn songs, and who to mate with, right? B is incorrect because it's agonistic behavior, and so that's a type of innate behavior. It's social behavior related to fighting. D is incorrect because it's an example of migratory behavior. And E is incorrect because the behavior is an example of fixed action pattern, right, which we just had an example of before. So it's unchangeable, and once it's initiated, is carried to completion. So C, hydra initially contracting when gently touched and then soon stopping to respond is our correct answer. 33, by convincing soldiers that they are a part of a brotherhood and thus increasing the likelihood that they will protect and even die for each other, the military is, consciously or unconsciously, tapping into genes that were adaptive in our ancestors due to kin selection. B is the correct answer here. A is incorrect because individual selection selects separate organisms based on their own phenotypes. C is incorrect because neoteny or neoteny, depending on how you say it, is one of the two ways by which a patomorphism can arise. And so that's the retention of embryonic features as an adult. So like with the axolotl, right? It actually retains its gills. D is incorrect because there's no such thing as inclusive selection. There is inclusive fitness, but not inclusive selection. E is incorrect because heterochrony is developmental change in the timing of events, leading to changes in size and shape. And so that doesn't really fit. So B is really what it's acting on here. 34. A certain plant you are studying has alleles big R and little r for pointed and round leaves, and alleles B, big B, and little b for blue and white petals. You perform a cross between the homozygous dominant and homozygous recessive plants. The offspring are then crossed with homozygous recessive plant. You observe the results. 1,600 offspring are produced. Which of the following data is closest to what you would expect given a recombination frequency of 25%? So here you can go back to Campbell chapter 15 if you need to. The frequency of recombination is a measure of the degree to which recombinants are found following crosses between specific parental types. If two loci are not linked, then the frequency of recombination will be equal to 50%, meaning that half of the progeny will be recombinants because of independent assortment. If two loci are linked, the frequency of recombination will be less than 50%. And so to calculate the frequency, you can use the formula frequency of recombination equals number of recombinants divided by recombinants plus parental types. And so if we're told that there is a recombination frequency of 25%, that means that 12.5% of offspring will have the big R little b chromosome, and 12.5% will have the little r big B chromosome. 37.5% will have the big R big B, and 37.5% will have little r little b. And so if we actually do the math here, right, then we actually get E 600 with the homozygous recessive, 200 with homozygous recessive for R and heterozygous for B, 200 homozygous R, nope, heterozygous R, homozygous recessive B, and 600 of the heterozygotes for both here. And you can rule out these other ones. Just this is one of those where it's actually kind of nice if you could just follow a formula, right? Then it will not lead you astray. In birds, the keel is an extension of the sternum where the powerful pectoral flight muscles attach. Which of the following orders of birds is most likely to have a reduced keel? So the keel is an extension of the sternum where the powerful pectoral flight muscles attach. Which of the following of birds is most likely to have a reduced keel? It would be one that doesn't fly, right? So in this case, A is the group for woodpeckers belong to that group. B, waterfowl belong to that group. C, penguins belong to that group. And they technically, you could say they fly through water instead of air. E is where chickens fall under. And so D is actually things like an emu here. So here you're just going to have to remember which group, right? Which ones are flightless and which ones are not. And you can go back to chapter 34 in Campbell for that information. What provides the best evidence for the independent origin of microphylls and megaphylls? So microphylls have a single unbranched vein, yes, while megaphylls have a branching vascular system. That's true. B is like the opposite. So if you remember from chapter 29 in Campbell, right, what the difference between microphylls and megaphylls even is. C, microphylls are small, where megaphylls are larger. It doesn't have to be that way. Sometimes, yeah, not always. We have two tiny examples there in the picture. Both are vascularized. The independent origin, that would be more of a, if they have the same trait, right, that's more of an ancestral trait, and you would imagine that they evolved together and not independently, which is what the question is asking for, is independent. E is incorrect because both serve the same function. So it's probable, right, it's the same as D in the fact that that means they would have evolved together instead of independently. So A is our best answer here. A male child with Klinefelter syndrome is found to be colorblind, which is a recessive X-linked trait. Both the mother and father have normal vision. How can this be explained? Right? So if you have a recessive X-linked trait, but both the mother and the father have normal vision, this is most likely, so since fathers don't transfer X chromosomes, right, you can rule out C and D, right, because they pass on the Y chromosome. So there's two out. The mother's genotype should be X big A, X little a, because she and her husband are both healthy. So that means she's got one recessive, right? But the other one is normal. So if a non-disjunction occurred during meiosis 1, then all tetrads of sex chromosomes would go to one of the cells. And the gametes will either have 2 copies of the same chromosome or none at all. And so then that means that the gametes can have 2 X chromosomes or 0 X chromosomes, which if they had 0 X chromosome, it would be an unviable embryo if fertilization were to occur. So since the mother is a carrier, then the son of each meiosis 1 non-disjunction would actually not yield the result. So the son would not have the disease. So therefore, the non-disjunction had to have occurred in meiosis 2 because during interphase 1, the egg cells would have 2 copies of the normal chromosome and 2 copies of the abnormal, right? 2 copies of the recessive diseased trait. Because during S phase, right, when DNA duplicates, so after meiosis 1, 2 new cells are produced, each of which has either the 2 recessive disease traits, right, or they have 2 normal genotypes that won't lead to disease. If non-disjunction occurs in meiosis 2, that means, right, like we're seeing here, that you'll get gametes that are either homozygous recessive, right, and diseased, or homozygous dominant, not diseased. Should they be fertilized with a sperm, then the resulting offspring would be either homozygous recessive X plus the Y chromosome from the male, or homozygous dominant with normal vision plus the Y. So statement E is incorrect because normal males X innovation, inactivations. Statement A is incorrect because in normal males, no X inactivation occurs. And so if it does, the male has a chromosomal abnormality. So we knocked out C, D, and E, right? And A now, so B is our correct answer. 38, birds and mammals share all the following characteristics except. So in this case, C is the correct answer. Statement A is incorrect because the amniotes are a group of tetrapods that have amniotic eggs equipped with 4 extra embryonic membranes. Statement B is incorrect because the notochord is a flexible rod-shaped organ, which develops from mesoderm and is found in embryos of all chordates. D is incorrect because both mammals and birds are endotherms. And E is incorrect because birds and mammals have a 4-chambered heart. And if you need to go back to brush up on this, it's chapter 42 in Campbell. Which of the following is not true about allopatric speciation? Doing a quick check. Okay, make sure the test and the slideshow match up every now and then. So allopatric speciation is more common than sympatric speciation because it's when a geographic barrier separates species and makes their genomes change over time, right? To more rapidly evolve, since species will have to then adapt, right, to new environments on either side of this geographic boundary. And so A is correct, right? Because that is actually the definition of allopatric speciation. B is incorrect because, like I just said, allopatric is more common than sympatric. C would be sympatric speciation, could be caused by that. Let's see. It can prevent, let's see. So it can be caused by natural selection under no, because it's caused by a geographic boundary, right? So D is incorrect. E, it can prevent interbreeding with the parent population. No, I'm trying to think of like the logistics there, but basically that's not, I mean, it's not the correct answer. It can prevent interbreeding with the parent population. I mean, it's by a geographic boundary, right? So you've got a geographic structure that pops up. So that doesn't really apply or make much sense for that one. So B is definitely our correct answer here. 40, which of the following is not true about genetic drift? So if you remember, genetic drift is one of the main factors that can cause allele frequency fluctuations in a population over time. And in doing so, right, it can cause harmful alleles to become fixed. It can also cause allele frequencies to change at random. Drift and mutation are really only our only random causes of allele frequency changes. C, it is significant in small populations, definitely. And this is all actually just part of the general definition of genetic drift. D can lead to a loss of genetic variation. Absolutely. This is where we get like the bottleneck effect, right? When we have extinction, like cheetahs, right? Their population went through a population bottleneck, and cheetahs are not very genetically diverse anymore due to that. E prevents allele frequencies. No, so genetic drift causes them to fluctuate. It doesn't prevent them. So E is our best answer. 41. An individual with Turner syndrome would have which of the following 6 chromosome arrangements? This is where you can actually go back to chapter 15 in Campbell and look at aneuploidy would be the term here. Turner syndrome is the only viable monosomy found in humans. And so this is a case where you have one chromosome only, right? And you're missing the other one. So A here, YO is actually non-viable since there's no X chromosomes whatsoever. So an embryo, right, that was fertilized, right, by a sperm. So an egg that had this, well, wait, yeah, no, if you had an embryo with the, it's like, wait, hold on, egg wouldn't have a Y. Anyway, if you had an embryo with this genotype, it wouldn't survive. And so A is our answer. None of the, notice that B, C, and E are not examples of monosomies, right? E is a trisomy. B has an extra Y here, and then C, you've got a a chromosomal duplication, so you've got a tetra something. And like I said, Y wouldn't, YO or Y0, right, wouldn't be viable because there's no X chromosome whatsoever. But A, you can actually have, that's the only viable option. 42, when the base composition of DNA from bacterium, Fusobacterium novum, was determined, 32% of the bases were found to be guanine. What is the pyrimidine content? So here, right, pyrimidines are T's and C's, and purines are A's and G's. So if you have 32% is guanine, that you have 32% of C. And that means that that is 64% of the genome, right? And so that means you've got the other 36%. Nope, what did I just say? Yeah, that's right. 36% is going to be A's and T's, which means 18% will be A's, 18% will be T's. So then if you take T and C, right? T and C are pyrimidines. Then you're taking 32% plus 18%, and you get 50%. And this is from chapter 5 in Campbell. 43, from which geological period over 500 million years ago did fossils first show all of the major body plans we see today? And so this is the Cambrian period, if you remember the Cambrian explosion, right? I'm not going to go through all the rest of these, but if you need to brush up on this, it's Campbell chapter 25. 44. Robert MacArthur and E.O. Wilson studied island biogeography and developed the island equilibrium model. Which of the following scenarios will definitely result in an island X having greater equilibrium number of species than island Y? So this is from chapter 54 of Campbell. The larger the island and the closer to the mainland it is, the more species will live there due to greater immigration rates and decreased extinction rates. Larger islands have more space than smaller islands, so there are more likely to be more niches and resources available. And extinction will be lower on island close to the mainland because of greater likelihood of immigration. And so extinction rates are larger on small islands. So because we've got island X, which is bigger, that's kind of its winning characteristic, I guess, is just that it's bigger, even though they're both the same distance, but since it's bigger, there's more niches that can be filled. B, island X is farther from the mainland. So that means it definitely wouldn't have any. C, island X is bigger than island Y and farther from the mainland, right? We need it to be closer. We need it to be bigger and closer. And so D or E, neither of those have that option either. So A is our correct answer here. And if you need to brush up on this, like I said, it's chapter 54, and this may look familiar to you, hopefully. 45, almost there. Which of the following is not an adaptation to survival in a grassland biome? Grassland biomes are large areas of grasses, flowers, and herbs, if you remember. And it's a region where the average annual precipitation is large enough to support the growth of grasses and maybe even a few trees. But it's unpredictable, the precipitation is, and it fluctuates. And so grasses have basal meristems that allow them to grow from the base of the plant instead of the tip. So that's true. And that's called intercalary meristems. Oh, excuse me. C, grasses have deep roots to anchor them to provide starch reserves. And yeah, that's true because when a fire or predators damage the upper parts of the grass, it needs to regrow from the starch reserves. Statement D is true because grassland biomes are quite devoid of water, and plants must tightly regulate the opening of their stomata to prevent dehydration. Okay, statement E is also true because in grassland biomes, there are almost no trees or other tall plants. And so in order to avoid predators, grazing animals have perfect vision to be able to see danger. So B is our correct answer here. And here's just a reminder of, it's got all of these breakdowns for you in chapter 52. My dog just knocked something over. I'm not sure if you heard that. 46. In many bird and mammal species, more often males have secondary sexual characters like the antlers of the deer or colored plumage in birds, and or perform elaborate courtship displays in order to attract females for copulation. Why do you think females are so often said to be the choosy sex? So here, if we read through all of these, which I'm not going to do in the interest of time, E is our correct answer. And if you need to check it and check why, you can go back to chapter 51. 47, males of a small tropical bird species spend 6 months of the year jumping around in a small area on the forest floor, exposing their bright yellow beard during landing. Males don't contribute anything other than sperm to the production of their offspring. Which of the following statements best explains why males display themselves in such an obvious manner that would also make them easy prey? So here, A is our correct answer. And B is incorrect because males that display more frequently and faster tend to obtain more, not less, matings. C is incorrect because a male's appearance is an indirect evidence of good male genetic quality, important for healthy offspring. D is incorrect because tropical biome is the species' richest place on Earth. This means that it has huge variety of different predators. Statement E is incorrect because the displays decrease an animal's chance of survival due to bright yellow color being more conspicuous and attractive to predators. So bright colors may make an animal prone to attack. Okay. Sexual selection is a case of natural selection that describes evolution due not to variable survival rates due to fitness, but rather due to variable reproductive rates stemming from characteristics that allow an individual to successfully attract a mate. These traits are called secondary sexual characters. Which answer is not a correct statement about these? And so reproductive organs are secondary sexual characters. So secondary sex characteristics are features that appear during puberty and sexual maturity, especially those that distinguish the two sexes of a species. So things like enlargement of breasts, growth of body hair, widening of hips, face is more rounded, smaller waist. In males, things like growth of body hair, muscle mass, facial hair. These are distinguished from primary sex characteristics, which are sex organs, right? So that's why B is our correct answer. And you can check out chapter 46 in Campbell if you need to review. 49, you observe an animal in the early stages of development. The zygote divides radially into 8 cells. If you separate these early cells, each one can successfully develop into an animal on its own, and the coelom forms from an infolding pocket of the archenteron, depending on which way you say it. Which of the following is a valid candidate for the animal you are observing? This is a sea urchin. So with this one, so the rest of this doesn't seem to really fit necessarily. So, but you can look at things like radial and indeterminate division, right? Those things are indicative of deuterostomes, which include echinoderms and chordates. And so here, right, you just have one of those options. Okay, and you can go back to chapter 32 if you need to review in Campbell. Last one. Which of the following classes contains organisms lacking a closed circulatory system? So from all of these, polychaetes are worms, which have a closed circulatory system. Cephalopods are the only mollusk with a closed circulatory system. Reptiles and eutheria have closed circulatory systems. So C is our correct answer here. All right, I'll see you guys at 1.